Good morning. No, it's good afternoon already, isn't it? Well, good afternoon, everyone. Thank you very much for joining us for this coffee seminar. For those of you who don't know me, I'm Ian Godwin. I'm the Director of the Centre for Crop Science in coffee. And if I could have the next slide, please. So UQ acknowledges the traditional owners and their custodianship of the lands on which we meet. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognise their valuable contributions to Australian and global society. So next slide, please. So as you're all aware, today's seminar is scheduled to finish at 1 p.m. And after the presentation, we're going to hold a question and answer session inside that envelope. At any time during the webinar, you can type your questions in the Q&A tab. Please make sure that you put them in the Q&A tab, not the chat function, or we're unable to use those questions. Next slide, please. So it's with great pleasure to be able to introduce Dr. Peter Crisp, who I know quite a few of you know. But for those of you who don't, Pete did his undergrad and PhD at the Australian National University in Canberra. He's a native of Canberra. He's one of those people who managed to escape from the place and probably, unlike the people who go there holidaying on government money, he's probably going to be someone who will actually contribute to making the world a better place. He then left Australia and became a postdoc at the University of Minnesota. He was in the Nathan Springer group working on maize genetics and epigenomics. And in 2019, he interviewed for a position in the School of Agriculture and Food Science. And he was successful in getting that position. And he decided that he was going to join in February, March 2020. In the interim, he had also applied for an ARC DECRA fellowship, and he was successful in getting that fellowship. So when he started at UQ, he effectively went on to that DECRA fellowship, which was a wonderful thing for both him and for his school, because they saved a lot of money because they didn't have to pay his salary. Pete and I share a lab in the John Hines building, where his group is growing at such a rapid rate that I think he'll probably kick me out in the next few years. That's just the way things go. So without further ado, I'm going to turn it over to you, Pete.
Alrighty. I think you should be able to see me now. So thanks, Ian, for the very nice introduction. It's been fantastic sharing a lab with you and your group. Thanks to the organisers for inviting me to talk today. I too acknowledge the traditional owners and their custodianship of the lands in which we meet today, and pay respects to their ancestors and descendants. So, a bit of background about me. I think Ian already introduced some of this. So as Ian said, prior to UQ, I was at the University of Minnesota, where I was working mostly on maize in Nathan Springer's lab. And so Minnesota is really a fantastic place, so well worth a visit if you get the chance. It has many claims to fame, for example, Norman borlaug, as well as some occupational hazards, such as snow during harvest. This was the last season that I was there, and winter came early, as you can see in our cornfield. As Ian said, prior to Minnesota, I was at ANU in Canberra, an ANU graduate. So it was quite fortuitous that I got the position in SAFS and started at UQ in January of last year, right before the pandemic really took hold. So it's been a bit of a crazy ride since then, but not only is Brisbane really a fantastic place, so it's been terrific to be here writing out the pandemic rather than somewhere else in the world. But so here at UQ, we have a research group. We study crop epigenomics and biotechnology. And so we span both wet lab and computational, so bioinformatics. We work on a range of plant species, so all the way from the biotech powerhouse, Benthamiana, through to cereals like sorghum, wheat, barley, maize. We do a mix of fundamental research in kind of molecular genetics, genomics, epigenetics. We also have a range of projects with applied goals, for instance, improving grain quality in cereals. So here, just up front, I wanted to acknowledge the team this year. So we're really off and running now, and these are the people who are driving the research forward. And you're going to hear about a couple of their projects today. So I just wanted to say thanks to the great team who really got our research going here over the last year. Okay, so today this is a talk with two parts, and these two parts really kind of reflect two of the major research directions in the lab. And there's a review on the screen there where we kind of summarize these two main directions and a lot of the concepts I'm going to talk about today. So if you're interested, you could have a read of that if you like. All right, so what is epigenetics? So here are a couple of examples. So on the left, there's 3 pairs of plants. You can see each of these pairs is quite phenotypically different. So flower morphology, tomato ripening, and fruit in oil palm. Now these plants, these pairs are each genetically identical, and the differences in phenotype are caused by epigenetic differences. And so just to give you a bit more of detail about one of these examples. So the bottom one is oil palm. And now oil palm can be propagated through tissue culture to maintain clonal varieties. But what can happen is that when these plants come out of tissue culture, they can develop this so-called mantled phenotype. So this manifests at fruiting, and essentially the yield is ruined because of a mantled phenotype. And it turns out that the basis of this phenotype is epigenetic. So a particular epigenetic mark called DNA methylation, it's altered by the tissue culturing process at a particular gene that's important for flowering, and that ends up ruining the yield of these plants. So that's a very important example, especially in the context of biotech. So this is one more, not yet famous example, I guess. So this is some wheat plants. This is a collaboration we have with CSIRO. This is some of their published work. They took wheat and treated it with an epigenetic inhibitor. And you can see this caused a range of different spike phenotypes. We're working to understand the basis of some of these phenotypes. All right, so there were some examples, but now I want to introduce you to two of the key definitions of epigenetics. And so firstly, there's what I would call the strict definition. And so here, the definition is that epigenetics refers to the heritable transmission of information that's not solely due to DNA sequence. And so the key here is heritability. And so this could be through cell division or across generations. But the broader definitions, this is the one you've probably more likely encountered previously or in the literature. This broader definition really just refers to the study of chromatin and the regulation of gene expression. And so in both of these examples, one of the keys is the modification of chromatin modifications. So these could be chemical modifications to DNA or histone modifications. But the important thing is that these chromatin modifications are a mechanism, can be a mechanism of epigenetic memory, but are not necessarily epigenetic on their own. And today, the key chromatin modification that I'm going to focus on is DNA methylation. So this is chemical modification to the cytosine in DNA at the 5' position. All right, so what could be the utility of epigenetics in agriculture? So for a very long time, we have selected phenotype based on phenotype in most species, and it's worked very well. And so where we have selection based on phenotype, the molecular basis of traits kind of didn't matter. It could be genetic or epigenetic. But now we have advanced tools such as genomic selection, which can allow selection based on genotype. So you could select or predict the best performing individuals or even the best parents to cross based on their genotype. And so this kind of raises the question, and this is one of the questions we're really motivated by, given these. So the question is, could the methylome provide additional information that would potentially be missed by these SNP-based selection schemes? So if we're selecting on genotype, could we now start to be missing important heritable epigenetic factors? Alternatively, and this is kind of the second question, relates to the second part of this talk, could we use epigenetic data, in particular methylation, to help us focus on the functionally relevant portions of the genome? Now, so a good portion of our research is fundamental in nature, it's basic science, but we're also aiming to help with a really big problem that the world is facing. And so here on the left, this is a graph of the increases in maize yield in the US over the last century. And you can see there's been several really big step changes in the rate of gain in maize yields. And these have been enabled by breakthroughs in breeding technology, mostly related to the use of hybrids. And this has been an amazing achievement. And actually that line kicks up a little bit more now due to the latest kind of molecular and gene editing innovations. But here's the problem: despite these, really tremendous advances, as you can see on the right, on our current trajectory, we're just not going to make enough food by 2050 to feed the world. And so what does this mean? I mean, this is obviously a complex problem. We're making great advances to continually increase yield. But I think what this does mean is that we need additional new avenues to help us speed up the rate of gain. And we think that epigenetics could help. So we study a variety of kind of the fundamental aspects of epigenetics in crops. We're aiming to understand these processes to determine if they could be useful in crop improvement, perhaps in pre-breeding or in breeding. And so we have, in this diagram, we have quite a number of projects focused on the concepts up the top. For instance, we're using CRISPR to knock out enzymes that regulate DNA methylation and epigenetic processes, to study that and to also create new variation. We're studying the effect of various breeding technologies like tissue culture, their effect on the epigenome, chemical treatments. But what I really wanted to mention briefly today is the prospects of population scale epigenomic profiling, which could be used for selection and breeding. So that's really the funnel in this diagram, kind of the key step to master to determine if this could be useful in breeding. So now, one of the reasons that epigenetics and DNA methylation is so exciting, and also the reason it could be useful, is because that it does not always abide by the principles of Mendelian inheritance. So there's this potential that it could contribute to so-called missing heritability. And there's also the possibility of the transmission of acquired traits. And so how might this be useful in agriculture? And so this really relates to the stability and heritability of DNA methylation. And so if we have a scale here of stability or heritability, where on the left we have very unstable, all the way to the right, very stable. Now, if DNA methylation and epialleles are very unstable, they're obviously not going to be very useful for breeding, because you're not going to be able to select for them. On the other hand, if they were very stable as genetic variation, they're also probably not very useful, because they would already be captured by SNP-based scans. If we could determine the genotype, we'd be able to infer the epigenotype. On the other hand, if they had intermediate stability, then they really could be of utility. And in fact, that's what we think. So I haven't explained yet, but DNA methylation comes in several flavors, depending on the sequence context the methylation is in. And we call these CHH context, CG and CHG. And the reason that's significant is that CHH methylation, which is a minor component of large crop genomes like maize, is relatively unstable. It's quite variable. On the other hand, CG and CHG type methylation is actually, on the whole, quite stable. It's not as stable as genetic variants, but it kind of occupies this space here, which really does open up the possibility that it could be useful for breeding. And in fact, this is what we have found when we've looked in maize. So this was a study that, a great study that I was fortunate to be part of. It was led by a former postdoc of Nathan's, Ching Li. And in this study, we profiled DNA methylation in a maize population. And the take-home message was that we could identify a large number of epialleles that were associated with traits. So this was an epigewos, but these were not associated with SNPs. So they wouldn't have been captured by a SNP-based scan. This was quite exciting, but in this study, we actually used the maize genome is quite big, and we used a sequence capture-based approach, which only profiled a small percent of the maize genome. And so perhaps you're wondering, if DNA methylation, the epigenome, has this great potential, why isn't everybody sequencing it? Why hasn't it been incorporated into breeding programs? And really, one of the key reasons for that is actually cost. It can be quite expensive. So to give you one example, using the current gold standard whole genome approach, whole genome bisulfite sequencing, if you did that in wheat, it's going to cost you probably more than $10,000 for one sample. And so it's really not feasible to do that on a population scale in crops with large genomes like wheat, barley, maize. Now, there are alternatives out there, including the sequence capture approach we used in maize. There's also reduced representation approaches, but these have drawbacks. So if you're doing a sequence capture, you need to know ahead of time the regions that are interesting. And if you're doing reduced representation, you're randomly sampling the genome, you're quite likely to miss the interesting bits. And so we've been thinking about this question quite a lot, or this challenge, this problem. And since moving to UQ, we've been working on a few tricks to kind of address this problem, and with the aim of dramatically reducing the costs of profiling methylation. And we've been making some exciting progress, which hopefully I can tell you about next time. So watch this space. But it seems like things are going actually very well. And so we've started on a set of quite ambitious projects, which, counting it up yesterday, realized that we're actually aiming now to profile methylation in over 1,000 varieties across several target crops. And so this includes sorghum. So this is a great collaboration we have with some of the folks in coffee. So Emma, David, and Ian. And in our group, PhD student Udas is spearheading these efforts. And so our aim there is to essentially do an epigewos in a sorghum diversity panel. We're also currently doing this in wheat and planning to do it in barley as well. And that's working with Lee Hickey and Kai, who's now in Germany. And this project actually is also in collaboration with the UQ Genome Innovation Hub. So I wanted to give a shout out to them. They've really been instrumental this year in helping us get this up and running, and we couldn't have got so far without them. So thanks, guys. And Judy is going to be tackling barley in the coming year. Another species that we're quite interested in, is grape. Grape being interesting because it's largely grafted, clonally propagated. And so this area of research, this line of research I'm very excited about, and it really promises to be a big year ahead. All right, so that's all I had to say about part one. And so now moving on to part two, where this second part is really in the area of that second definition of epigenetics, where we're not necessarily thinking about inheritance, but we're using epigenome profiles to understand the genome. Now, this was a project that started in Nathan's lab in Minnesota and then has continued here at UQ, and it's also the work that's partially funded by my DECRA. And so again, in this aspect, in this part, we're mainly focusing on the chromatin modification, DNA methylation. All right, so to return to thinking about why this all matters, why does chromatin and epigenetics matter? And so chromatin epigenetic regulation has several key functions related to, say, genome integrity, the control of transposable elements, but a really important one is the control of gene expression. So chromatin plays a major role in regulating the expression of genes, and really this is what gives rise to the complexity of life. So here you can see the cross-section of a leaf. You can see there are many different cell types. And this complexity is enabled by the precise control of gene expression, which in turn is regulated by chromatin. So here, I just wanted to give you some of the quick recap on the highlights of gene expression. So here we have a gene. Now, the transcriptional level for this gene to be expressed, it needs to be transcribed by an RNA polymerase. That's going to bind to a core promoter, but we know that's not the whole story. This core promoter is essential, but not sufficient usually to drive expression. What we also need are cis regulatory elements. So these are DNA sequences that bind to other cofactors like transcription factors. And so these cis regulatory elements, the important thing is they could be found right adjacent to a gene, so in the promoter region, but they could also be found hundreds of kilobase pairs away from the gene they regulate. And so when that's the case, what actually happens, as you can see in this diagram, is that cis regulatory element, when it binds the right transcription factor, loops around and interacts with the promoter of the gene to drive transcription. And so the important point here is the activity of these regulatory elements, their availability to bind transcription factors is regulated by chromatin. So if they have repressive chromatin marks, they can be compacted and inaccessible to the transcriptional machinery and to transcription factors, whereas if they have permissible marks, they can become open and active. And so this is another way of thinking about that same thing, the activity of cis regulatory elements. So in this diagram, this is from maize. Most of this section is about maize. And so this is an expression map of one particular gene. So anywhere it's red, that gene is expressed. Anywhere it's yellow, that gene is essentially off. So this particular gene, you might notice, is very specifically expressed in the shoot apical meristem and also in the embryo. And so you can imagine if this is the gene, this gene might have two nearby regulatory regions. And so when a particular say a meristem transcription factor comes in and binds to the first regulatory region, we get chromatin looping, and that can drive expression specifically in the meristem. Alternatively, perhaps now a repressor comes in and binds to that region, and a new suite of transcription factors might bind the second regulatory element. This leads to a new looping conformation, and this is what could drive expression in the embryo. And now we know this kind of regulation is not only important, but also valuable in agriculture, and we can look at domestication to get a flavor of that. And so here on this slide, I'm showing two key domestication traits from crops. So we have non-shattering and also plant architecture. Now for non-shattering, if we take maize as an example, this domestication trait, non-shattering, was driven by a change in the sequence of a key gene. Whereas in sorghum and rice, the genetic change which drove this domestication trait, or which enabled this beneficial trait, was in a regulatory region outside the coding sequence of the gene, so either in the promoter or upstream. And we see the same thing for plant architecture. For example, in maize, key architectural change during domestication, going from the progenitor teosinte to modern maize, was driven by a change in a regulatory region that was actually 60 kb upstream of the gene it regulates. Now, everyone's probably heard of gene editing and the promise of CRISPR-Cas9 for controlling or generating new traits in crops. And so this is conventionally used to target a gene, to create deletions, and essentially, or commonly, to knock out the function of the gene it's targeting. Now, the problem with this conventional approach is sometimes a gene knockout can be deleterious or even lethal. But now, knowing the value of regulatory variation, we can actually, and people have done this in this example in maize, target regulatory regions. So here in this example, this is targeting one of the key genes regulating meristems in maize. So we have this feedback loop, two of the key players being Wuschel and clavata3. And so what was found is in maize, if we knock out the clavata3 homolog, we do actually get a slight increase in cob size, but overall it's detrimental to yield. But if rather than targeting the coding sequence of the gene to generate a knockout, if we target the promoter of the gene, what can be achieved is rather than a knockout, we can knock down, reduce expression or change expression patterns. And this was found to actually overall increase yields in maize. And so also one of the key points here is by targeting the promoter, you can create a variety of alleles and actually generate quantitative variation for this trait. So not just small or big, but a range of sizes for the maize cob. So here's one of the problems. In crops with large genomes, the cis regulatory elements, while they're sometimes in the promoter, they can also be very far from the genes that they target. So the classic example, again, being this gene, so TB1 in maize, where the key regulatory change occurs 60 kb away from the gene that it's regulating. This raises the question, how do we find or locate these regulatory elements in a large crop genome? And so one of the technologies that has emerged over the last few years is chromatin accessibility. So this can be, as a particular method using a transposase. Essentially, we can extract chromatin from a plant and then tag, using a hyperactive transposase, any regions that have accessible chromatin. And this has proved to be a very effective method to identify cis regulatory elements that are far from genes. So this is data from maize. So this regulatory region for TB1 in maize, which took many, many years to identify using classical genetics, we can now rediscover that same region using a TAC-seq in several days. And in fact, when people have profiled accessible chromatin across the entire maize genome, they discovered this really quite incredible statistic that even though open chromatin makes up less than 1% of the genome, that 1% captures a nucleotide variation in that 1% explains more than 40% of phenotypic variation across traits. So these open and accessible regions are where most of the functionally important variation is occurring. But a TAC-seq also has this problem. And so that first track I was showing you was chromatin accessibility from leaf tissue. Now if we profile a second tissue, so now we're into the ear or the inflorescence, we can see that this first region has actually closed and a second region has opened up. And so what this means is that it works very well if you're interested in one specific tissue, but if you wanted to discover all of the cis elements in a genome, you're going to have to profile a huge number of combinations of tissue, environment, genotype, etc. And so thinking about this problem, we had the idea, what if rather than using chromatin accessibility, we could use methylation to discover all of the cis elements in a genome. This wasn't exactly a new idea because people have known for a long time that regions that have accessible chromatin also have low levels of DNA methylation. But our idea was kind of to flip that approach. So rather than starting with the accessible regions, could we just profile methylation to discover cis regulatory elements? And so this is what this looks like. So this is a methylation profile of a region of the maize genome. So the colored bars represent methylation, different colors representing the different flavors that we mentioned briefly. So you can see as you go along, we have methylation and then there's a gap. And this particular gap with a bit of blue flavored methylation, CG methylation, is a typical pattern of a gene. As we go along further though, you can see there are 4 regions that lack methylation and we call these unmethylated regions, UMRs. Now, if we compare this to our gold standard TAC-seq chromatin accessibility profiling, we can see in the same region, there are 2 regions with open chromatin and we capture both using methylation alone. But this was the big question for us. So yes, we capture the known regions, but then we have 3 additional unmethylated regions that in leaf tissue are inaccessible. So we call these inaccessible UMRs, IUMRs. The big question for us was, could these unmethylated regions that were not accessible in leaf, could they be functional? Could they actually encode for cis regulatory elements? So that was our big question. And so so I've already mentioned how chromatin accessibility is quite dynamic. That's one of its drawbacks. If we compare 3 different tissues, we can see that accessibility varies quite dramatically. But the reason we're quite excited about using methylation is what we've seen many times is that methylation is actually very stable during most of plant vegetative development. So if we compare a leaf and a root, the regions that are unmethylated are very, very stable between those 2 tissues. So, and here, so we can see a profile of a leaf and a root, and you can see just qualitatively, these profiles are incredibly similar. So this got us thinking, what if these regions that lack methylation in the leaf, but are not accessible, what if they could be marking a region that could become accessible in another tissue? And so, so that's what we investigated. And so here is an example of another classic loci in maize. This is, so here we have RAP 2.7, it's a gene, it's important for flowering time. And it was known to also have a regulatory element. Again, this is quite far from the gene, 70 kb from the gene. It was a QTL map using classical genetics. It's known to affect flowering time. Okay, so we profiled DNA methylation in the maize leaf. And again, we identify several unmethylated regions upstream of this gene. Here I've highlighted 4 of them. So the question was, which ones of these are accessible? So when we look at accessibility in leaf, we can see this first one is indeed accessible in the leaf. But then we have these 3 additional regions, lack methylation in leaf, but are inaccessible, so their chromatin is closed. Could these open in another tissue? So sure enough, when we looked at root, we can see here now that region 1 and region 3 have opened. So DNA methylation in the leaf had marked these regions and they open in another tissue. Now we still have region 4 here, which wasn't open in either leaf or root, so perhaps that opens in another, a third tissue, which we haven't profiled yet. So here is a second example. This is another, this is TB1. I won't go through this one, but it's just to say it works for other examples. So those were 2 specific examples. Now on a global scale, if we take all of the unmethylated regions in the maize genome and we say how many of them are accessible in the leaf, we capture 99% of them. If we go to a second tissue, now root, and take all of the regions that are opening in root, how many of them were predicted from the leaf? Again, we predict almost all of them. Third tissue, ear, and you can see if we were to continue profiling different tissues, we think we would essentially fill in this circle. So that diagram was for regions close to genes, and we see the same trend for regions far away from genes. Now, we did a number of additional studies that I'm not going to go through today to investigate whether to look for more evidence that these UMRs could be functional. And we did, in fact, find that they are involved in chromatin looping, they're enriched for transcription factor binding. And when we do reporter gene assays, they seem to be able to also drive expression of reporter genes. And so all of that work was in maize, but excitingly, this isn't just restricted to maize. It works in other species. One of the interesting things we've seen when we go to other species is that even though, so here we're profiling several grass species that range dramatically in genome sizes, but the total size of the UMR space is actually incredibly conserved around, in absolute terms, it was very similar across these species. And so actually we've done several other species now, and we're currently working to get some of this data online. So if you were interested, you would be able to go and look at your favorite gene. But, so we think, so what does this mean? So we think we've got this method essentially that would enable you to profile DNA methylation in a single tissue, an easily accessible tissue like the leaf, and predict all of the cis regulatory elements in a genome. And you could imagine that could be very useful as we start to use gene editing to create new traits or to create new trait variation. But really where we're at is that we think this method has promise, and now we're trying to further validate it and also apply it to some of our favorite crop traits. And so Kimi is driving some of this research forward, in particular in Benthamiana, with help from Jimmy and Peter's group. And so we're working firstly in Benthamiana so we can test this on a number of targets. And essentially what Kimi is doing is picking a range of target genes and then trying to edit the unmethylated regions that we predict are important for controlling the expression of those genes. And she's using some really exciting new technology where we can essentially do gene editing by avoiding tissue culture. But we're also now trying to do this in sorghum and also barley. And so Haley, as well as Kimmy, is trying to do this same approach with some of our favorite genes in sorghum. And this is working with Ian and his group, so Karen and Lou. And I thought I'd tell you very briefly about one of the genes we're interested in sorghum. And so, Ian's group for a number of years now has been interested in grain quality in sorghum, and they have a range of genes they're interested in. But one particular gene was of interest to us because what Ian's group has found is that so they had a gene. It was implicated in sorghum grain quality, and so first they made an RNAi line. And this actually was very beneficial for quality traits, grain quality traits in sorghum. High yield, grain size, protein content, and digestibility. Of course, the drawback being it's RNAi, so it's GM. So now that we have CRISPR-Cas9 technology, gene editing, the idea was to recreate this using CRISPR. The obvious advantage there being you could then segregate away the transgenes and have a non-GM version. But somewhat surprisingly, when they made the CRISPR line, although it maintained some of the beneficial traits, overall it had a yield penalty and altered development. And so our working hypothesis is that actually what we want for this gene is to knock it down or to alter the expression of it rather than knocking it out completely. And so you can imagine that's a really good candidate for this approach we're using, where we might be able to identify regulatory elements to target for CRISPR to change the expression level or pattern of this gene. And so this is what we've been working on, and we have in fact found for this gene some unmethylated regions. In this example, they're actually relatively close to the gene. But one of the really cool things is we found these unmethylated regions that we think could mark cis regulatory elements. And we had the idea to look at these in maize to compare whether they've been conserved. So the cool thing in maize is, so for this gene, as you might know, after sorghum and maize split, maize underwent a whole genome duplication. So for many genes, there are still 2 copies. And this gene, that is the case. There are 2 copies. They've both been maintained. But the interesting thing is these 2 regions that we identified as putative regulatory regions, they're both conserved. They've been maintained in maize. However, each of the maize genes only retained one of the regulatory regions. And the even cooler thing is, if we then look at the expression pattern of these 2 genes in maize, one of them is now expressed in the embryo, whereas the other one is expressed in the endosperm. So now we have this really neat hypothesis that not only have we identified 2 regulatory regions, but based on synteny with maize, we predict which one of them is actually controlling expression in the endosperm. And so now Kimmy and Haley are working to clone these regions, study their expression patterns, but more importantly, to gene edit them to see what that does to, in sorghum, to see what that does to grain quality. All right, so thanks for listening today. I'd really like to acknowledge the lab and all the great work everyone's doing, our collaborators, in particular, everyone that's been very incredibly generous and welcoming here at UQ, notably, of course, Ian, sharing a lab with, and our funders. Thanks.
Thanks very much, Pete, and thanks for letting me share your lab too. Because one thing we don't forget is that on paper it is listed as a SAFS lab. So there we go. It's a great example of the 2 organisations, the 2 things working together. We do have so there's a question from Michael Advardi. So Michael's question is, if UMRs are open for business, what makes them inaccessible in different tissues? are you able to isolate them and explore what molecules may be bound to them to make them inaccessible?
Yeah, great question. Yeah, I guess the answer to that is it's the usual processes that regulate, the opening and closing of chromatin. I think actually the interesting question is, if these regions are inaccessible, what maintains their lack of methylation in other tissues? Yeah, so it's the usual, chromatin regulatory pathways. But yeah, potentially we could isolate these and look at what's being bound for sure.
So, Pete, I was going to ask. So, from a point of view of outside effects on changing methylation patterns, or basically changing the epigenome, do things like plant pathogen interactions or temperature or light have a large known effect on these types of things?
Yeah, so a couple of important points. The epigenome can be quite dynamic if we're talking about histone modifications, chromatin accessibility. But if we're talking about DNA methylation, it's relatively stable. So there are some treatments, some conditions that are known to lead to changes. Actually, the kind of condition or the stress that is best known for affecting DNA methylation is tissue culture. And there are some examples: salt stress, perhaps heat stress. But on the whole, I think those should be considered exceptions. Most of the time, environmental pressures, DNA methylation in particular, seems relatively impervious. It's stable through those. But yeah. Okay.
I also was wondering, especially when you were showing those chromatin loop structures, is there any evidence that differences in methylation patterns, etc., actually play a role with larger genome reorganizations that effectively become genomic, like translocations, inversions, those types of things?
Yeah, definitely. So people have found that patterns of DNA methylation affect chromatin interactions. But another example is, people in the crop space are being quite interested in methylation because it's known to affect recombination. In plants that have altered methylation, mutants in the methylation pathway seem to have altered rates of recombination. And from a breeding perspective, that could be quite interesting or useful if you could alter or increase rates of recombination. So yeah, definitely.
Okay, I've got another specific question about the example that you gave with the sorghum grain quality. So you've got two potential candidates in sorghum for that gene. Now, and seeing that the maize gene has only got one or the other of those, but at least that shows that the maize gene is going to keep that function. Whereas if you knock out or alter one of those UMRs in sorghum, it hasn't got a redundant copy that can perhaps do things that are necessary. Do you feel that it's that there's a potential there that you're going to find out that that it works, but it also has some sort of adverse effect on sorghum plant growth or development or something?
Yeah, it's certainly possible. From what we've seen from some of the examples for other genes, other groups have tried, for instance, using gene editing to make deletions in promoters. It seems, if you make a variety of different alleles, you can generate a variety of different expression patterns. It is possible that there's one key element, and if we knock it out, we really severely affect the expression of this gene. But I mean, based on what's been done for other genes, we're quite hopeful that we could generate new expression patterns or a variety of levels of expression. I think there's good precedent that it could work. Of course, we don't know. We have to try and see what happens.
Okay, so we've got one from Peter Waterhouse. Do you see any conserved sequence motifs in UMRs?
Yeah, so for sure. So I mean, one evidence for conserved so I guess there's two ways you could look at that. We can look at transcription factor binding. So we see, if we look at a particular transcription factor, we see enrichment in these unmethylated regions. So there's definitely enrichment for those short motifs. We have looked at conserved non-coding sequences, for instance, between sorghum and maize, and we do see enrichment in the unmethylated portion of the genome. Yeah, so definitely.
Okay, so we've got the questions seem to be coming thick and fast now, just as I was about to wrap up. So I'll try and keep the question short. So Lee Hickey's asked if futuristic genotyping platforms for plant breeding can capture epi-alleles, what would the data look like? And would it capture expression levels so that you could apply selection for particular expression patterns?
Yeah, I mean, so that's really our hope. So, you know, it's definitely possible that, so if we're talking genotype, you know, I guess that data type is you have a SNP or you don't, and we could essentially convert, for instance, methylation data to a similar data type. You have a particular epi-allele or you don't, you know. In its raw form, the data is quantitative, so it gives you a percentage of methylation, but it's definitely possible to then convert that into a methylated, unmethylated, or indeterminate criteria. And so, yes, definitely that's what we're interested in. You know, could we profile methylation and then use that as extra information for either selection or even genomic prediction? Maybe it accounts for a portion of heritability of a trait, or maybe it could improve prediction accuracies. Okay,
I think we've got time for one more. So we've got a question from Owen Powell, who says, you mentioned missing heritability. Has anyone demonstrated that epigenetics fill the gap in estimates of genetic variation?
I think that's probably an open question. I mean, certainly when we profiled, you know, the population in maize, we found epi-alleles associated with traits that were not captured by SNPs. But I don't know that you know, there aren't that many studies that have done this well, mainly because of the cost of doing those studies. But hopefully in, you know, in the next years to come, we'll have some data to really test that properly.
Okay, thanks, Pete. Well, in the interest of time, thanks again very much, and I'm sure everyone else is clapping too. If we could just advance to the next slide, that would be great. Oh yeah, well, we've already done that, so let's go to the next one. So just to remind everyone that next week we've got our next seminar in the Coffee Science series. It's Dr. Anthony van Heerwaarden, who's talking about using a physiological framework to solve production issues in tree crops. And the final slide just reminds people that you can view all the Science Seminars webpage here, and to remind people also that these are recorded and you can get links to those recordings so you can watch them, and you're welcome to send them out to other people to watch as well. So thank you very much, everyone, and I hope you enjoy the rest of your afternoon, and thanks again, Pete.